formation of a seed is a fascinating and intricate process that marks a crucial stage in the life cycle of higher plants. From pollination to seed maturation, this journey involves a series of meticulously orchestrated events that ensure the continuity of plant species. In this exploration, we will delve into the intricate steps of seed formation, highlighting the key biological processes that contribute to the development of a mature seed. The journey begins with pollination, a process where pollen grains are transferred from the male reproductive organ, anther, to the female reproductive organ, estigma, of a flower. This can occur through various mechanisms, such as wind, insects, birds, or other pollinators. The goal is to facilitate the fusion of male and female gametes, ultimately leading to the formation of a fertilized ovule. Once a pollen grain lands on the stigma, it germinates and develops a pollen tube, a slender structure that extends down the style towards the ovary. This tube serves as a conduit for the male gametes, sperm cells, to reach the ovule, housed within the ovary. The journey of the pollen tube is guided by chemical signals, ensuring precise navigation towards the target. Upon reaching the ovule, the pollen tube penetrates the ovule's protective layers. This penetration marks the beginning of the fertilization process. One of the sperm cells fuses with the egg cell, forming a diploid zygote. This fusion is the essence of sexual reproduction in plants, combining genetic material from both parent plants to create a unique genetic makeup for the offspring. As fertilization progresses, other cells within the ovule also undergo fusion, leading to the formation of endosperm. Endosperm is a nutrient-rich tissue that provides nourishment to the developing embryo. This collaborative effort between the egg cell and other cells sets the stage for the initiation of seed development. The fertilized ovule, now referred to as the fertilized egg or zygote, undergoes a series of divisions and differentiations, ultimately giving rise to the embryo. The embryo represents the young developing plant within the seed. Depending on the plant species, the embryo can have one or more cotyledons, seed leaves, and consists of a radicle, embryonic root, plumule, embryonic shoot, and hypocotyl, the region between the radicle and plumule. Simultaneously, the ovule undergoes structural changes, transforming into a seed. The outer layers of the ovule develop into the seed coat, a protective layer that shields the embryo from external threats. The seed coat also plays a crucial role in seed dormancy, a state of suspended growth that allows the seed to withstand unfavorable conditions until germination is favorable. As the embryo develops, so does the surrounding seed tissues. The endosperm, formed during fertilization, continues to provide nutrients to the growing embryo. In some plant species, the endosperm persists in the mature seed, serving as a nutrient reserve for the germinating seedling. The maturation of the seed involves the accumulation of storage reserves, such as starches, lipids, and proteins. These reserves serve as a source of energy and nutrients for the emerging seedling during germination. The composition and quantity of storage reserves vary among plant species, reflecting their adaptation to specific ecological niches and germination conditions. Seed maturation is often accompanied by changes in water content, which decreases as the seed dries. This reduction in water content contributes to the establishment of seed dormancy, a crucial adaptation that allows seeds to survive. Once the seed has completed its maturation process, it enters a state of dormancy, awaiting the right environmental cues to trigger germination. External factors such as temperature, light, and moisture play pivotal roles in breaking seed dormancy and initiating the germination process. When environmental conditions become favorable, the seed absorbs water, rehydrating its cells and activating metabolic processes. Enzymes within the seed facilitate the breakdown of stored reserves into simpler forms that can be utilized by the emerging seedling. The radicle, the embryonic root, emerges first, followed by the plumule, which develops into the shoot. As the seedling continues to grow, it relies on the nutrients stored in the seed until it can independently photosynthesize and sustain itself. The journey from pollination to seed germination represents a remarkable cycle of life, ensuring the survival and dispersal of plant species across diverse ecosystems. In conclusion, the formation of a seed is a complex and tightly regulated process that encompasses pollination, fertilization, embryo development, and seed maturation. This intricate journey ensures the production of viable seeds that can withstand adverse conditions and successfully germinate when environmental conditions are conducive to the growth of a new plant.